Hi, thanks everyone. My name is Richard Poiré. I am the manager of the ANU node of the Australian Plant Phenomics Network in Canberra. And yeah, again, it's a last minute feeling, so I'm happy to present some of what we do in Australia. We are the Australian Plant Phenomics Network, and we set ourselves to be the partnering with various organizations for your plant phenotyping needs. We recognize that it's quite a complex subject, and that's why we in Australia also have a very fragile ecosystem. And it's been identified as a strategic direction 15 years ago that we need a network of coordinated facilities to tackle this challenge and provide our scientists and breeders with the right tools and the right expertise to do that work. So we used to be known as the Australian Plant Phenomics Facility, but as of this year, we are changed to the Australian Plant Phenomics Network because we went from 2 to 9 facilities, which warrants the change in structure and name. You can see on this map where the facilities are located. So we cover pretty much the entirety of the territory. And we have facilities that specialize in control environment phenotyping, 7 out of 9 of the nodes. We have facilities that specialize into the field phenomics, and we have central facilities also that help us collaborating together. We are collaborative by design, and it's not just a good intention. We also have people that are dedicated to make sure that we do collaborate within the nodes, but also outside. And for instance, data management is something that is tackled at the central level. So we have a team of people that make sure that the data we collect in every node can neatly interface with each other to enable meta-analysis and other work so that we make sure that the data is reusable. It takes a village to do all of this work, and it's not just my work that I'm presenting here. So this is our team, for instance, at ANU. As of last year, we have a new director, Danny Way. Unfortunately, she could not make it to this meeting, but she came from Canada to start a position as a researcher, but also works as a director for the node. I am the manager, but then also within the team, we have expertise within project management, horticulture, biology, computer science, mechatronics, and biostatistics. It takes very much all of the skills to make it all work. And plant phenomics is quite complicated. I think the talks this morning showcase that. And we don't expect anyone to have all of the answers, but we provide a seamless experience by working together on that. So I will present some of the core offering that we are presenting, This is the very first plant I phenotyped a bit over 20 years ago. So it was slightly destructive. The plant didn't recover that, but I was harvesting plants, taking pictures of them, and then by hand measuring leaf length, width, and it's time consuming, gives you good data, but at the end of the day, it was not fast enough for the type of work we were planning to do. So we had to evolve towards more advanced technologies. And I think that's what plant phenomics has done over the last 20 years. And to be able to have a good phenotype, I think it all starts with a good control, a good environment that we can either control or monitor. So in our case, it's a collection of growth chambers that we have, like in different configuration that allow for different level of climate control, from the control, like the growth chambers, to the glass house, to more recently, we invested into growth capsules that are retrofitted shipping containers that have the latest technology in environmental control, whether it's temperature, humidity, multispectral LEDs. And some of them even have 3D multispectral scanners. And we also have tools that allow us to probe into the physiology of the plant. We have thermal camera, chlorophyll fluorescence systems, and hyperspectral cameras and scanners that allow to look into more complex traits. So some of the chambers that we have, they cover ranges there from -10 to +40 degrees. So Australia has pretty extreme climate, and if you want to reproduce some of those climate conditions indoor, we need to have pretty strong control over that. Our facility is both PC2 compliant. That means that we can make and work with GM organisms, but we also have a facility that is quarantine compliant, which is very important. Australia is very protective with how it deals with quarantine laws. And if you want to import plants and work on quarantine material, it requires a certain level of containment that we do provide. We also specialize in dynamic climate control. So we do have rather intricate way to control the chambers. We don't just program like a day and a night temperature. We have ways to model the environmental conditions and plug that into the chamber so that you have the full dynamic change in temperature, light, and humidity at the 10-minute interval based on GPS coordinate of a condition, like a location we'd like to replicate, or artificial scenario when we want to induce environmental stress. We also have glasshouse space that people are able to access. We can control CO2, we can elevate it, and essentially make the plant grow into conditions that they are likely to experience in the next few decades. And we also have attached lab space that we can do PC2 work next to them. And more recently, we have 8 of those outdoor growth chambers, some of them with very high light capability, some of them with multispectral capability, and 2 of them with multispectral scanners that we can use to monitor the canopy growth over time. But it's not just the environment. We also have like quite a bunch of portable equipment that people can take from us and do some measurements with. One of the workhorse of our technologies, for instance, at the moment has been the use of portable hyperspec equipment. People use that in the field, in the lab. And 2 years ago, I was presenting a model that we've done on how to use this system to predict photosynthetic capacity at a much quicker rate than what we were able to do previously. We also have access to various hyperspectral scanner instruments. We have 2 of them at the moment. Chlorophyll fluorescence systems that we keep up to date regularly, but also coming from the physiology and ecophysiology side, we also have access to a range of gas exchange systems to measure plant photosynthesis. More recently, we've procured pyrometers that people can deploy in the field, and they're very easy to use, and they do both the pyrometry and the fluorescence in pretty much one click. And thermal cameras that we've been using quite extensively. And again, it's not just the kit, it's really the expertise. And when people come and reach out to us, they often have a scientific question but not know which tool to use, which one would be the most suited for their scientific question. So I think it's been a journey with them to make sure that they have access to the right tool, the right expertise, and design the experiment properly. And yeah, there's a lot that we also do with in the recent years with the 3D phenotyping of the plants. Most of the plants that we phenotype these days are hopelessly 3D, and just a top-down picture of the plant is not good enough to resolve the architecture of the plant. So that's why we've been investing in 3D canopy scanners, and this is the first Phenospec system that we had here. We have a second one now with the more modern sensors. They work very well, and they've been quite absolute workhorse of our recent experiments. They allow us to measure plants up to 1.2 meter in height, and the container that they are growing in goes all the way to 2.1 meter. And they generate both the point cloud and the CSV file that people need for their publication needs. So this is one example of the point cloud that can be generated on wheat plants. It's highly resolved, like we get sub-millimeter accuracy on those, and we can collect traits based on two fields. Both we have a range of structural traits that revolve around biomass, plant height, leaf area, and a 3D leaf area. And other parameters, but we also have spectral traits that are more linked to the color of the plants and how we assess their greenness, the way we can bin the pixels into the pale green, the light green, the yellow, the brown, and also a range of indices based from the remote sensing community. But we also do work in the field, and I think I can't understate like just how much work it takes to coordinate a team of people working across Australia. So we provide field logistics. We've been training teams of people doing physiological measurement in the field. More recently, there's been a GRDC heat tolerance campaign that we've been coordinating. So it really involves training up all of the people involved in the acquisition of data in the field to make sure that they collect the data the same way, they have good experience on the instrument that they're working with, and coordinating the field and working in the field with various weather conditions. Field campaigns that have to be postponed occasionally is quite hard. But yeah, our team has been doing very well there. And a couple of case studies of work that we've done in the past. We have worked with a ag-tech company who makes plant-based vaccines, so growing vaccines into plants using recombinant transformation of the plant. And they approached us to see whether we could find ways to better determine the yield predictors, so how to assess how much protein there is in the plant. And that protein is completely invisible, and unless you assess the plant by destructive measurement, you have no idea of how much protein of the compound of interest you have in the plant. So we've designed this high-resolution platform to reconstruct quite faithfully the architecture of the plant that they're working with at a leaf level. So we have leaf segmentation, that algorithm that we've developed to can identify the plants, and we've been able to track essentially and the history of the plant through the phenotype, and being able to identify which ones would be the most suited for the subsequent transformation, with the intent later on to use this 3D mesh as a way to map the hyperspectral traits that we've been developing. And another project that we have going on at the moment is as part of Australia's Economic Accelerator. Molecular farmers really need tools to be able to better assess their protein yield in plant. And one of the projects that we've been developing this year is testing a range of construct and transformation in the plant and phenotyping them with a hyperspectral scan to identify and quantify how much protein we have in the plant at different time points. We're still training the model at this point. We are still having to do the destructive measurement, but by the end of the year, we'll have a trained model that we'll be able to predict just by the hyperspectral signature how much protein of interest we have in the plant when it grows. One of the emerging directions that we are taking at ANU is also in looking into the hidden path and the root development of the plants. So we've procured 100 rhizo boxes. So they are pretty tall. They're like 1.2 meter tall rhizo boxes. So we have them already, but now at the moment we are designing the frame and the cassette that will be enabling us to do the phenotyping of those. So it's being fabricated at ANU in our workshop at the moment by the team of engineers. And within the next few months, we'll have a working system where we can water the plants and take out one by one. It's a semi-automatic system. It's not a fully automated one, but allows us to work with for rhizo boxes. We are looking into structural traits, but we're also looking at doing hyperspectral imagery of the plant and being able to collect exudates and have a bit of a better idea of how the physiology of the roots and how they're developing underground. But we also do develop a lot of custom software solutions that we are more than willing to share with people if they're interested. We've noticed that a lot of our users were doing a lot of that manually themselves, and we thought that there has to be a better way to do it. So our software engineer developed a first tool called Leaf Analyzer that replaces the need to use flatbed scanners or a conveyor belt system for the measurement of detached leaves. So it's based on a target that has QR codes attached to it. So all it takes is a smartphone or whichever camera of choice you have to take a picture of those. And the software does automatically the segmentation of the background, the foreground, when it works with very difficult lighting conditions and report automatically about like the type of metrics that plant biologists are usually keen to get. So this is an example of what it looks like. So the left side is the raw pictures, and the right one is geometrically corrected and has the background removed, and it automatically saves the metrics, the color, the size on a CSV file that people can use. So yeah, that software is ready to go, and if people are interested in using it, we're more than happy to share it. It works with leaf, but it works with pretty much any material you can think of. It any plant organ that is not white will be segmented pretty decently with it. Works with also to count objects, and it really takes no input from the user, so it's very fast and very user friendly. Another recent development that we've done is using regarding the segmentation of objects, and especially when you're looking at hyperspectral imagery, there's not much in terms of tools available to work in hyperspec dimensions. So we, Tao in our team created this software that allows us to segment pretty much anything with no click if it's very easy, or with a few pointers if the image is quite complex. So let me load a demo of how the system works. So by loading an image that was previously recorded, and all we need to do is identify essentially a region of interest that so some cropping within the image. The system will do a first guess of what the object of interest might be, and in that case, it's doing a good job at identifying what we want. But it's not. Sometimes it's not perfect the first go. So what Tao is doing here is placing a few pointers about like, yes, this is what I'm interested in, are the green dots, and the red dots are the one that, no, this is not exactly what we wanted. So the system will automatically correct the prediction and account for the user input when they are required. It depends on the complexity of the image really. And working with complex root images, sometimes it's very useful to have those, and automatically then it will save all of the metrics of the objects that have been collected there. So we intend to use this software as a first pass to segment the foreground, background in our riser boxes. So this is an example here, and it works very well even with very complex soil and architecture. We're in the process of improving that even further. And the last piece of software application is with regard to hyperspectral imagery. As I mentioned, there's very few software that allows to do that. So what we have here is a plant that has been scanned through our hyperspectral scanner, but usually it's quite difficult to work with those images. So what we have here is a single click essentially to identify where the plant object is. The system will make a guess of what the plant is, and it will propagate the mask onto all of the hyperspectral layers and save the image as essentially a binary masked hyperspectral file that will allow for very much easier subsequent work and predictions on all of the pixels. So essentially each pixel becomes its own full hyperspectral scan, and we have a range of hyperspectral prediction model based on those data. So and that cleans up the data and speeds up the work a lot. So I think that's pretty much all I had this morning to present. But yeah, by essence, we are highly collaborative. So everything you can see here works for us, but our facility is really looking forward to collaboration with academics, industry, both within Australia and internationally. So if you have any interest, I think there's quite a few of us here. There are 6 of us from APPN. So yeah, reach out to us, and I'm sure we can find ways to work together. Thank you.
Thanks, Chuck. Do you have any questions? Good.
Hi, thanks for presenting the work you do on the other side of the planet. And a question regarding the segmentation tool you showed. is there also an option to have, for example, if you have loads of images, like hundreds of thousands of images, that you can pre-select and kind of train it or do a profile that you then apply as in an automated pipeline as a batch processing?
Yeah, it does batch processing. Yes.
I just thought, is it based on one of the segmentation tool?
Is the segmentation tool based on something like one of those meta tools or something like that that's available, or did you all make something?
No, I mean, those are using pre-trained models, but we package that into something that is very, very easily usable.
Okay. And then was it trained specifically to work better on in this use case, this sort of use case, than if you just took the plain?
No, it works pretty much with any image. Actually, a lot of our team members use that to do snapshots of things on the internet and grab logos and things to copy paste. Like it really works on anything. It's agnostic. It's not just plants. Works on anything.
Yes, thanks for the talk. And I was indeed also interested by the software which you developed. How does this relate to the backbone of, for instance, something like Plant CV? Can you share this through a Jupyter notebook or a Plant CV backbone such that we all can try and take advantage from it?
No, we would be very happy to open source it and make it part of Plant CV. Like especially, I think the hyperspec module is quite novel, so I think there's value in having that. It came from like the specific needs that our user had, so we developed that. But we realized that there's more wider appeal for this kind of software. So yeah, happy to discuss how we can do that.
Yep, I could do it. So we are also working on some segmentation tools, some labeling tools, some AI training environment. It's not a question, it's more a suggestion. We got this new IPPN AI working group. I think we should align on what we are doing, because it feels a bit that we are doing similar things in parallel, and we don't need to have the best tool. We should have a common tool and speed up the developments.
Yeah,
Do you agree?
I do agree, and I think for me, one of the highlights of coming to this meeting is seeing just how many networks of infrastructures we have, but like sometimes it's very hard to work collaboratively. We all want to be collaborative, but I think within APPF we are fortunate enough to have like a central team that makes sure that we are collaborative. But when we are talking about networks of networks, like what's the vehicle to make sure that operating procedures are saved somewhere, accessible for people? Where do we find software that people can use and reuse? More than happy to explore what that could look like.
Thank you for your presentation. My question is this software only working for leaves threads, or we can segmentation fruit, flower, and nodes, and another part of the plant?
Yeah, any organ. It's not optimized for leaf. No, it's it works for leaf, and it will generate parameters that are meaningful for like length and width that usually make sense for leaf tissue, but would work equally as well fruit, as long as they're not white.
Okay, thank you so much.